when we were talking about free energy, I said that temperature is the average kinetic energy of a system. Cells are a system and they have a certain amount of temperature, which means the molecules inside the cell have certain amount of kinetic energy, which allows them to move and bump into each other. So our skeletal elements, they have to overcome that thermal barrier, which is basically the energy, the kinetic energy in the system, which can cause these skeletal elements to dissociate or disintegrate or depolymerize. So let's see how these things tie up together. The cytoskeletal polymers are strong and adaptable because they are built from multiple protofilaments. Long, linear strings of subunits join end to end that associate with one another also laterally. So, strength and adaptability. The basic principle is making these structures from smaller units. Protofilaments basically are the filaments that ultimately form the larger filamental structures. I will show you a protofilament, what I mean by protofilament, in the next slide. But smaller units or smaller strings of these monomers, they twist and turn around each other in a helical lattice. I will also show you a diagram of what I mean by helical lattice. We have seen another molecule which has a helical structure, that is DNA. So if there is a string of these polymers, the string of these monomers, they form the polymer in a linear form, and other such polymers, they can twist and turn around each other, stabilizing the larger structure, which is basically our cytoskeletal element. The addition or loss of a subunit at one of the ends of protofilaments makes or breaks one set of longitudinal bonds and either one or two sets of lateral bonds. So what I'm saying is like the drawing I made in one of the previous modules. So the subunits, they can associate with each other longitudinally and also laterally to form larger filamentous structures. So this is the lateral association, and this is basically the longitudinal association. So addition or loss of a subunit, so if we are to add a subunit here, it will result in formation of either longitudinal bond or binding force, or it will be also, if we were to add another subunit here, so it will form a lateral and also longitudinal interaction with the existing polymer. So if you lose one or one monomer at one end, protofilament of a protofilament, longitudinal or lateral bonds have to be either broken or formed. In contrast, if the composite filament we were to break it in the middle. So, for example, if we were to break this in somewhere in the middle, this will result in breaking of more associations or more bonds, those weaker interactions or weaker bonds, than if we were to lose at the very tip. I hope that is clear, but I'll again show you in a proper diagram what I mean by that. So let's look at a more proper diagram in the next slide. So here again we have a string of monomers attached with each other because of these weaker interactions. So you can easily break the string in the middle since you're only dissociating one bond, one attractive force or one type of bonding entity that is holding this structure together. So if you were to or breaking this filament in the middle or removing one monomer at the very tip, they're about the same amount of energy required to break it either in the middle or just removing one monomer from the very tip. A filament formed in this way would not have enough strength to avoid breakage by ambient thermal energy unless subunits were bound very tightly to its neighbor. So if there were more of these subunits here, there were more bonding between these subunits and the lateral subunits, the structure will be more stable, thermally more stable. Let me show you the next slide, what I mean by that. Here, for example, now if you look at each of these green balls, which is basically representing a single monomer, this, for example, in the middle is being held in its place by 4 of its neighbors. So 4 forces are holding this single monomer. In order to break it laterally, this filament laterally, we will have to get rid of a lot of these bonding forces, whereas if we were to just break one of these monomers at the very tip, it will require that we break one longitudinal bond, or 2 or perhaps 1 or 2 perhaps lateral bonds. So again, it basically making protofilaments and laterally associating them increases the thermal stability of our cytoskeletal elements. So large energy differences between these two processes allow most cytoskeletal filaments to resist the thermal breakage. If our filaments were not thermally stable, they would not be able to perform their function, which is one of their function is, of course, providing the structural support to the cell. However, leaving with this type of arrangement, it allows these filaments, these structures, to still be flexible, because it is easy to remove monomers from the tip. So you have a stable structure, but yet it is dynamic, meaning that it can be reorganized, it can be broken into smaller parts from the tip, and they can assemble at a different location. So cytoskeletal subunits are held together by a large number of hydrophobic interactions and weak non-covalent bonds. We have talked about it. For example, hydrogen bonding is also a non-covalent weak bond. The location and types of subunit contexts are different for different cytoskeletal filaments. I'll show you in the next slide what I mean. We have just seen a structure which was closer to microtubule. Now I'll show you closer. We will see in more detail how intermediate filaments, for example, the monomers are locked in place. Intermediate filaments, for example, they assemble by forming strong lateral contacts between alpha helices and coils which extend over most of the length of each elongated fibrous subunit. And these individual subunits are staggered in the filament, so intermediate filaments can tolerate stretching and bending forces, so they are more like a rope. Let me show you this. So here we have intermediate filaments. So first of all, these monomers, they have strong lateral, there are a lot of lateral bonds forming between monomers. This one bar is basically a single subunit of intermediate filament. And as you can see, it is forming significantly more lateral associations than we saw earlier in case of, for example, those balls we talked about. And additionally, they are staggered, meaning that not all of them start and end at the same spot. So if they're staggered, this basically staggering makes them more stable, gives them the stability. So there are more bonds holding these monomers together. Microtubules built from globular structures held together are primarily held together by longitudinal bonds. Lateral bonds hold 13 protofilaments together, as they are comparatively weak as compared to intermediate filaments. So we will see we'll talk about in more detail how intermediate filaments and microtubule subunits come together and form these larger structures, but that will be in the next module.